So far we've taken quite a bit of time to talk about what happens to glucose and how we can start processing glucose to acquire high energy molecules like ATP and NADH. So we've talked about glucose coming in from our diet and the way that our body absorbs that. And we've also talked about the glucose storage molecule referred to as glycogen. Now the breakdown of glycogen into glucose molecules, or the absorption of glucose molecules from the food that we eat, will ultimately lead to the production, after the process of glycolysis is completed, to the production of pyruvate. So this is our pyruvate molecule. It is a 3-carbon molecule. You can see there are lots of oxygen atoms and lots of double bonds of oxygen in pyruvate. And we're going to look at 2 different options for pyruvate to be metabolized anaerobically. So remember that anaerobic conditions are conditions that are lacking oxygen, and in the absence of oxygen, the maximal amount of energy that's present in the covalent bonds of pyruvate can't actually be harvested. So the cells that are utilizing the glucose have to take another means to deal with pyruvate once it's produced from the initial steps of breaking down glucose. So we're going to talk about fermentation in yeast, and we'll talk about what happens under anaerobic exercise in skeletal muscle. Now you'll notice that both of these processes are going to require the utilization of an NADH molecule. Now remember that NADH is a molecule another high energy molecule that we can generate from the energy that's stored in the covalent bonds of glucose. However, in this case, because we've got to deal with pyruvate in a way that isn't a best case scenario, we actually consume an NADH molecule in the process. So we're going to start just by giving you guys what would happen under aerobic conditions. We're going to look at one more step that's going to occur, and then we're going to stop the discussion at this point until we enter into chapter 23. Now, the metabolism of pyruvate under aerobic conditions is absolutely the best case scenario, because that's where we can utilize all the energy that's present in pyruvate and break down the rest of these glucose molecules completely into carbon dioxide and water. So our pyruvate molecule, you'll notice it does have this carboxyl group at the end of it, just the same kind of group that we have at the end of a carboxylic acid. Now our pyruvate molecule is 3 carbons long, but what happens under aerobic conditions is that this carbon, along with these 2 oxygens, is removed to form carbon dioxide. Now the remaining 2 carbons are going to be attached to a coenzyme A molecule. That's a molecule I introduced to you guys in one of the earlier mini lectures of this chapter. So this 2-carbon group called an acetyl group is attached to coenzyme A at the sulfur atom to produce acetyl-CoA. Now acetyl-CoA is a very important intermediate molecule. We're going to see the generation of acetyl-CoA when we break down lipids in chapter 17. But regardless of where it comes from, acetyl-CoA is going to be used in another metabolic pathway called the citric acid cycle to generate lots of high energy molecules and complete the oxidation or the complete breakdown of, in our case right now, a glucose molecule. Now under anaerobic conditions, this isn't an option, and our 3-carbon molecule has to go down a different path. Now one path that it can go down is what happens in our skeletal muscles. So the pyruvate is going to be reduced, and the C double bond O that's present right here at carbon 2 is going to be converted into the hydroxyl group that you see here. So we're taking a double bond O and we're converting that to a single bond O. Now that's going to require the high energy NADH molecule and a proton. And again, an enzyme is going to be needed to create this product. So we're adding an H here to the oxygen to make the hydroxyl group, and we're adding an H as a single bond to this carbon to satisfy this carbon's need for 4 oxygens, or I'm sorry, for 4 covalent bonds. Now this molecule is lactate, or it's also referred to as lactic acid, because it is a version of a carboxylic acid. So we still have the carboxyl group right here. Now again, this is what happens in our muscle, and this product, lactate, is what is referred to as a metabolic dead end, which means that the muscle cells will produce this molecule, but once the molecule is produced, there's no other use for that molecule in your muscle cells. So after you're finished with your strenuous exercise that produces lactate, your cells and your body as a whole is going to have to deal with the accumulation of this molecule, and we'll talk about that at the end of this particular section. So NADH is oxidized. We're losing this high energy molecule and it's being converted back in to NADH. So again, this is going to happen under strenuous exercise conditions where oxygen is being depleted in the muscles more readily than it can actually be delivered there. So that's going to cause there to be temporarily anaerobic conditions within our muscle cells. So the lactate is going to accumulate, and that's going to cause the muscles to tire, and ultimately it's going to cause quite a bit of discomfort or pain in the athlete. So how does breathing change as a result of strenuous exercise? If you guys think about it, you know that your breathing, the rate of breathing increases. Now, of course, there are going to be multiple reasons for that. One of which is going to be the attempt of your body to get oxygen to all the tissues that need it. So the breathing becomes heavy while you're undergoing that strenuous exercise, but it doesn't stop even after you've completed that strenuous bout of exercise. So you need to continue with that large delivery of oxygen, because once the muscles are finished contracting, they still have to overcome the production of that metabolic dead end that we've talked about. So we have to basically take that lactate and revert it back into pyruvate, which is going to happen in the liver. And again, we'll talk about that in the last couple of slides. Now, the other option for pyruvate, which happens in other kinds of organisms, is this idea of fermentation. So fermentation is going to happen in organisms like yeast. And so the pyruvate molecule is still being metabolized under anaerobic conditions, so condition where we lack oxygen. And this is where oxygen isn't utilized. And that's going to be the condition that we have with yeast. So yeast cells metabolize glucose under anaerobic conditions. Now what happens here is the release of a carbon dioxide molecule. So we still take our 3-carbon pyruvate and we remove one carbon, the carbon of our carboxylate group. We remove the carboxylate group to form carbon dioxide. Now that's going to cause us to basically end up with a 2-carbon aldehyde. Now you guys know that molecule as ethanol. The common name is acetaldehyde. So ethanol is what you guys know it as, and so the ethanol can then be reduced. Again, if we've got the reduction of a double bond O, then we'll have the oxidation of something else. So the the double bond O is converted to an OH, the hydroxyl group, and a proton is used to satisfy the 4th covalent bond of our carbon atom. And so this is going to convert our ethanol aldehyde to ethanol. And ethanol, as you know, is going to be the product of yeast. And again, the production of carbon dioxide is what allows bread to rise whenever you have yeast that are being used in that capacity. However, yeast can also be used in the production of substances like wine. And so in that case, we're capitalizing on the yeast and their capacity to make ethanol. So ethanol being the alcohol that's present in substances like wine. So this is the second of the two possible anaerobic pathways where pyruvate can be utilized after the initial breakdown of glucose. So again, this is our pyruvate molecule. In skeletal muscle, all 3 carbons of our pyruvate molecule will remain intact, and that will generate the molecule lactate or lactic acid. Now this is the molecule that we're going to think about a little bit more in the next couple of slides. So there's a cycle that's referred to as the Cori cycle, whereby your body will deal with the extra production or the excessive production of lactate from the metabolism of glucose. So remember that glucose is broken down to pyruvate, and then in the muscles, when oxygen is in inadequate supply, the muscle cells will use the pyruvate to make lactate. Now the only way that your body can deal with that lactate is to take the lactate and ship it back to the liver, where it can be reverted back into pyruvate. So we'll talk about that with this diagram. So in your muscles, the glucose molecules are broken down to pyruvate, and then under anaerobic conditions, the pyruvate is converted into lactate or lactic acid. Now the lactic acid, because it's a metabolic dead end in your skeletal muscles, it will need to leave your skeletal muscle and through your circulation be delivered to the liver. Now in the liver, the lactate can be converted back into pyruvate because the cells of your liver have enzymes that can accomplish that. Now in your liver, the pyruvate can be utilized to synthesize a glucose molecule. This is through the process called gluconeogenesis. This glucose can then be delivered back into the blood, where it can be taken back up by muscle cells or other cells of your body. Or if there is adequate glucose in the blood, that glucose can then be stored as glycogen. Remember that glycogen storage can occur either in the liver, which is used for delivery of glucose into the blood for all tissues, or glycogen can be stored in the muscles for muscle contractions at a later time. So this is a diagram that kind of summarizes a lot of what we've talked about so far with these first couple of stages of glucose metabolism. So polysaccharides that are absorbed in your diet are broken down into individual monosaccharides, many of which are converted directly into glucose. The glucose can then be utilized for energy to make pyruvate, or it can be stored in the form of glycogen. So again, this whole excess glucose into glycogen is going to be this part over here. We talked about that the last time. If it is used for energy, it's going to initially be broken down into pyruvate, and then under ideal conditions, the pyruvate will then be converted into acetyl-CoA, or the coenzyme A molecule that's carrying a 2-carbon unit. This acetyl-CoA molecule is then going to enter into the citric acid cycle that we'll talk about in chapter 23.